Okay, welcome back, everybody. Dr. Linden here. We are continuing with muscle physiology, and we are going to be getting into a topic of what are some muscle proteins that make up the sarcomere. So the sarcomere is made up of contractile proteins, regulatory proteins, and structural proteins. So the contractile proteins you heard me mention before are referred to as actin and myosin. Actin are thin, myosin are thicker. So you can see here to the right these yellow little spheres right here, right? This is actin, and myosin are these on the left-hand side. The regulatory proteins are troponin and tropomyosin. So if we look on the yellow, if we look on the actin, we see this kind of like rust-looking or orange-looking string or rope, if you would. One's here, and here's the other one. These string-like fibers are referred to as tropomyosin. And tropomyosin is held in place by these blue things called troponin. Okay, these are two other types of proteins, tropomyosin and troponin. Troponin is kind of like a staple. This staple is, and these staples are holding the string in place. They're holding it in place. And you'll see that as they're being held in place, they are covering what is referred to as myosin binding sites. These little black circles here that are covered and not fully exposed are called myosin binding sites. So again, the myosin binding sites are being covered by this regulatory protein called tropomyosin, and what's holding the tropomyosin in place is the troponin. Now, the myosin binding site is on actin, and the myosin binding site is waiting for myosin to articulate or attach to it, but it has to be fully exposed. So in this particular instance, since the binding sites are covered, there's not going to be any connection between myosin and actin. Okay, just want to lay down some groundwork there for you. Now, the structural proteins are titin, nebulin, alpha-actin, myosin, and dystrophin. Okay, these are the two that we're going to spend quite a bit of time looking at. There are some autoimmune conditions that do happen that attack, let's say, muscular dystrophy. Muscular dystrophy is autoimmune against the degradation of this structural protein, dystrophin. Okay, so we have the myofibrils. Myofibrils are composed of these microfilaments. We said some are thin, some are thick. The thin ones are actin, and the thicker ones are myosin. They're arranged in contractile units. The contractile or functional unit of muscle is called a sarcomere. So the sarcomere is composed of three things. These three things: contractile proteins, regulatory proteins, and structural proteins. Think of the regulatory proteins as the on and off switch, and the contractile proteins, which are actin or thin, myosin or thick. These are the two that are going to interact with each other to slide past each other. And again, these are the structural proteins that deal with the alignment of the muscles, the stabilization of them, their elasticity, as well as their extensibility. Now, actin is composed of an F protein, a G protein. The G proteins are important. The G proteins are those spheres on actin where the binding sites are. And then there's tropomyosin and troponin. So we look here, right? Each of these Each of these is a G protein. And you can see that's where the binding sites are. And then, of course, we have tropomyosin and troponin. Those are all related to actin. The tropomyosin is the molecule that extends the entire length of the actin, and it's covering the myosin binding sites. That's what we showed you. Troponin is going to hold the tropomyosin over the binding site on the G protein until it's time for a contraction. And we have to get into detail as to what is actually time for the contraction. Okay, so what is going to remove troponin to expose these binding sites? So we have to have something that comes in to remove the staples, right? Troponin are the staples that are holding tropomyosin down. We need something to come in, remove the troponin, so that the tropomyosin shifts and moves out of the way so that these binding sites become exposed so that myosin can attach to it. Well, guess what that is? That key that unlocks troponin, or the staple remover, is actually calcium. Where was calcium stored? In the sarcoplasmic reticulum. Okay, things are starting to make sense now. So here, now we have all these G molecules of actin. We could see tropomyosin, and we could see, in this case, the troponin is now yellow. So troponin is the staple that holds the tropomyosin in place. You see the binding sites are not exposed. They are covered. So when calcium comes in, calcium removes the troponin. It removes the staple, so that tropomyosin shifts out of the way. And when it does, the binding sites are exposed for myosin, for these myosin heads to attach. And you could see how many heads there are. These myosin heads are waiting to find these binding sites to create a contraction. Okay, so this is just another view on the upper right. You can see all the G molecules. We could see the tropomyosin. We can see the troponin holding it in place. On the bottom, we see myosin. And I want you to think of myosin as your elbow and your wrist and your hand. Okay, so let's say this is your elbow joint. This hinge right here is your elbow. Then right here, this hinge is your wrist, and this would be your hand. So if you look at bending your elbow, and then you can flex your wrist, if you can see that image in your mind's eye, then you understand how myosin is going to work. The elbow has to bend so that this moves up, and then when this moves up, it can bind to the binding site, and then the wrist bends and creates a flick to create the contraction. When that happens, this is a sarcomere at rest. Look at this H zone here. This is the sarcomere at rest, but here you can see the H zone is much smaller. That's because the contraction took place. All of these little bluish, purplish myosin heads latched onto actin in red and slid them closer together to create a concentric contraction. So here is the axon. Here is the synaptic terminal. You could see we have the acetylcholine neurotransmitter. Now, calcium has to rush in. When that happens, acetylcholine is released, and it's going to bind to the receptors here. This is going to be repetition, what I'm telling you now. So the acetylcholine moves into the synaptic cleft, binds to the sodium channel on the motor end plate. Remember, the motor end plate's on the muscle. When that happens, boom, it goes right down the transverse tubule, that invagination. The action potential, boom, the electrical activity, boom, right down into that transverse tubule. The transverse tubule binds and connects to the sarcoplasmic reticulum. What does the sarcoplasmic reticulum store? What mineral? Calcium. When calcium is released, it hits that sarcomere. Remember that entire junction with actin and myosin. What is the calcium going to do? It's going to remove troponin so that the tropomyosin shifts and the binding sites are exposed. Now we have the elbow here that bends, it attaches, and then the wrist joint here that flexes, and this is going to create a contraction. When the contraction ends, we have to stop it, so we go through these steps where in order to end this, we have to get rid of number one, which is that neurotransmitter, acetylcholine. So we remove acetylcholine with an enzyme called acetylcholine esterase. Anything that ends in ASE or ACE is an enzyme. So acetylcholine is removed by acetylcholine esterase. Acetylcholine esterase, now that it's gone, none of this stuff happens. You don't get activation. You don't get anything going down the T-tubule. You don't get the release of calcium. So calcium goes back into storage. If you don't have calcium stimulating the sarcomere to remove troponin so that tropomyosin shifts, everything goes back to its resting state. The contraction ends and you have relaxation. Okay, so this slide here purely goes along with these steps. So the steps that initiate contraction are here on the left side, and then the steps that end contraction are here on the right side. So at the neuromuscular junction, acetylcholine was released. We know that. It binds to the receptors. Remember the sodium channel. So you have acetylcholine binding, the sodium channel opens. We're going to get an action potential. It's going to spread right down the T-tubule. The T-tubule connects to the sarcoplasmic reticulum. It's going to release the calcium that's stored there. The calcium that's stored there is going to bind to the troponin, removing the troponin. Remember, it's the staple remover. And when that happens, the tropomyosin complex shifts out of the way, exposing the binding site on actin, and a cross bridge forms. The cross bridge is the connection between the myosin heads and the binding site on actin. The contraction begins as repeated cycles of cross bridge binding takes place, pivoting, which is at the myosin head, and then detachment occurs. Now that detachment is powered by the breakdown of ATP. So we need ATP in order to detach the myosin head from actin. And we know that because it takes energy. Like if you see a person who, let's say, forensic analysis of the dead body, they see the person in a state of rigor mortis. Everything's in a flex state. The wrists are flexed, the elbows are flexed, they're in a C-shaped curve, they find the body, they're in the state of rigor mortis, everything is in contraction. Why? Well, they're dead. And when you're dead, you can't make ATP, which means your muscles can't relax, they can't detach. Now, after a period of time, the body does release these digestive proteins that just degrades and breaks down all the proteins. But the detachment of the myosin head does take ATP. Okay, the action potential ceases or ends when the acetylcholine is broken down by acetylcholine esterase. Then the calcium is reabsorbed by the sarcoplasmic reticulum. Then the troponin tropomyosin complex returns to its normal resting position. There's no more cross-bridge interaction, so there's no more sliding filament theory, and the muscle contraction ends and the muscle relaxes. That is muscle physiology. Okay? Now, what events can take place at the neuromuscular junction that produce the action potential? Well, these are the things that we've already discussed, which is just review. Okay, these are the key steps that we mentioned. We said that calcium channels in the synaptic end bulb open, So you get this influx of calcium. That influx of calcium causes exocytosis of the neurotransmitter called acetylcholine to be released in the synaptic cleft. Then that acetylcholine is going to bind to the sodium channel that's on that motor end plate. That's going to cause excitation, activation, excitation, depolarization, right? It all means the same thing. What's going to happen is that message goes down the T tubule, the transverse tubule, it butts up into the sarcoplasmic reticulum. You get the release of calcium. If you don't want a contraction to take place anymore, then you have to break down the acetylcholine with acetylcholine esterase. Okay, let's take a pause here. That was a lot of information.